Hello everyone, welcome back to my YouTube channel. This is Sahana LK from the Force and Pleasure section. Today I'm here to discuss about the biology for engineering. In that, I chosen a topic of nucleic acid. Shall we go through a nucleic acid? Nucleic acid are a long chain polymeric molecules. The monomer, it is nothing but the repeating unit, is known as the nucleotides, and here sometimes nucleic acids are referred to as a polynucleotides. Deoxyribonucleic acid, this stands for DNA, and the ribonucleic acid, it stands for RNA, are the two major types of nucleic acid. DNA and RNA are responsible for the inheritance and transmission of specific characters from one generation to another generation. As shall we go through our DNA? It is nothing but deoxyribonucleic acid. It is a chemically, DNA is a composed of pentose sugar, phosphoric acid, and some cyclic bases containing nitrogen. The sugar moiety present in DNA molecules is beta into D into 2 deoxyriboses. The cyclic bases that have nitrogen in them are adenine, guanine, cytosine, and thymine. These are the four components which are present in the nitrogen. These bases and their arrangement in the molecules of DNA play an important role in the storage of information from one generation to the next one. DNA has a double stranded helical structure, where RNA is a single strand helical structure in which the strands are complemented to one each other. RNA. RNA stands for ribonucleic acids. This RNA molecule is also composed of phosphoric acid, a pentose sugar, and some cyclic bases containing nitrogen. RNA has a beta D riboses in it as the sugar moiety. The heterocyclic bases present in RNA are adenine, guanine, cytosine, and uracil. In the DNA, we discussed that the bases are present is nothing but adenine, guanine, cytosine, and thymine. In place, here RNA is replaced by the uracil. It is a fourth component replacement. And in RNA, the fourth base is different from that of DNA, as we discussed. The RNA generally consists of a single strand which sometimes folds back. That results in a double helix structure. These are the three types of RNA molecules, each having a specific functions called message RNA, ribosomes RNA, transfer RNA is nothing but mRNA, rRNA, and tRNA. Functions of nucleic acids. Nucleic acids are responsible for the transmission of inherent characters from parent to offspring. As we know that we have a similar characteristics from the parents to the children. These are responsible for the synthesis of protein in our body. DNA fingerprinting is a method used to forensic experts to determine paternity. It is also used for the identification of criminals. As we know, it has also played a major role in studies regarding biological evolutions and the genetics. Shall we discuss DNA vaccine for rabies? DNA vaccines are bacterial plasmids constructed to express an encoded protein following in vivo administrations and the subsequent transfections of the cells. Several facts explaining the interest in a DNA-based rabies vaccine approach. Here, first one is the protection from rabies is commensurate with the presence of adequate amounts of virus neutralizing antibodies, principally targeted against the rabies virus glycoprotein. The tools of recommended DNA technology allow facial cloning of the glycoprotein gene into suitable expression vectors which mediate efficient in vivo expression of glycoprotein. Second one is nanogram amount of a plasmid encoded protein in transfected cells can generate high affinity antibodies. In the production of the protein with transport cells facilitates native post-translation modifications and observations on issues associated with protein purification in vitro. DNA vaccines provide unique approaches for expanding the spectrum of human responses, such as by the employing multiple or modified antigens and targeted delivery to specific cells, types, or locations. Remarkable thermal stability of plasmids over 50 degrees Celsius may be a particular advantageous in Tropical climates. Remarkable thermal stability of plasmids as we discussed over a 50 degrees Celsius. Fifth one is generic nature of the production and the purification process of plasmid vaccines reduces cost and facilities development compared to conventional vaccines. A DNA rabies vaccine, it is a simplest embodiment. It is a new eukaryotic expression vector containing the rabies virus glycoprotein gene under a suitable promoter. The glycoprotein sequence of Pasteur virus is PV. Challenge virus standards, it stands for CVS. Evelyn Rocky Nikki abstinence ERA or street virus associated have been used by different investigators who evaluated DNA rabies vaccines. Also, significant progress has been made in the design of vectors suited for gene delivery in the recent years. Plasmid DNA based rabies vaccination is yet to witness significant commercial success or applications in routine rabies prophylaxis. Drawbacks relating to poor immunology and requirement of higher DNA doses in large animals remains unsolved issues in rabies DNA vaccination. However, current progresses in the field of vector design hold considerable promises for rabies DNA vaccine development. Insights into the roles played by the antigen presenting cells in the shaping of adaptive immune response and the benefits of targeted antigen delivery to these cells hold great potential in devising better strategies for rabies DNA vaccinations. Co-delivery with molecularly defined adjustants is another approach to needs further evaluation in DNA rabies vaccination. Advances in the field of chemical biology also hold importance in the development of delivery agents and adjustments for rabies DNA vaccines. It is felt that DNA rabies vaccination will sustain research interest penetrating to rabies, at least in the animal host, provides its current drawbacks are addressed effectively. Here, has now discussed about the DNA. Shall we go through RNA? RNA is the messenger RNA mRNA vaccine. A coronavirus disease in 2019, that is COVID-19, vaccine can prevent you from getting COVID-19 or from becoming seriously ill state or dying due to COVID-19. Each COVID-19 vaccine causes the immune systems to create antibodies to fight COVID-19. These vaccines use a harmless version of a spike-like structure on the surface of the COVID-19 virus called S protein. Messenger RNA. Here, the type of vaccine gives your cells instructions for how to make the S protein found on the surface of the COVID-19 virus. After vaccination, your muscle cell begin making the S proteins pieces and displaying them on cell surfaces. This causes your body to create antibodies. If you later become infected with the COVID-19 virus, these antibodies will fight the virus. Once the protein pieces are made, the cells break down into the instructions and get rid of them. The mRNA in the vaccine doesn't enter the nucleus of the cell where DNA is kept. Both the BioNTech and the Moderna are the first vaccine introduced to solve the COVID-19. DNA fingerprinting. Satellite DNA regions are stretches of repetitive DNA which do not code for any specific protein. These non-coding sequences from a major chunk of the DNA profile of humans, they depict the high level of polymorphisms and are the basis of DNA fingerprinting. This gene shows a high level of polymorphism in all kinds of tissues, as a result of which they prove to be very successful in forensic studies. Any piece of DNA sample found at the crime scenes can be analyzed for the level of polymorphisms in the non-coding relative sequences. After the DNA profile is traced, it becomes easier to find the criminal by performing the DNA fingerprinting for the suspects. Apart from crime scenes, fingerprinting applications also prove useful in finding an unclaimed baby by conducting a paternity test on the DNA. Here, the following are the steps involved in DNA fingerprinting. First, we should isolate the DNA. Then, digesting the DNA with the help of restrictions endonuclease enzymes. Then, next step is separating the digested fragments as per the fragment size by the process of electrophoresis. Bloating the separated fragments onto synthetic members like nylon. Hybridizing the fragments using labeled VNTR probes. Analyzing the hybrid fragments using the autodiography. Has this much we have discussed about the DNA and the RNA are the nucleic acids. Thank you.